This is Chapter 6, Part 3, which is on enzyme regulation. How are enzymes regulated? There are many ways, including the synthesis and degradation of enzymes. There is the availability of substrates. So if there's more substrate around, the enzyme is more active. If there is less substrate around, the enzyme is less active. There is the localization or compartmentalization of enzymes in a cell. So if you keep an enzyme in a specific organelle, such as the mitochondria, that activity will stay there. There's product inhibition, feedback inhibition, allosteric effectors regulating allosteric enzymes, and covalent modification. So we'll go into more detail of 4 through 7 in the following slides. Product inhibition is when high levels of the product of the enzyme inhibits the enzyme. For example, coenzyme A, which is a product of an enzyme called histone acetyltransferase, can inhibit the enzyme activity. So histone acetyltransferases are enzymes that bind acetyl-CoA and proteins called histones as substrate. The histone acetyltransferase catalyzes chemistry where it transfers the acetyl group onto the side chain lysines of histone protein. So the acetyl group is attached to the lysine of the protein, forming acetylated histone protein and coenzyme A. Coenzyme A is a competitive inhibitor of histone acetyltransferase enzymes. The way that coenzyme A works is it actually outcompetes acetyl-CoA from the active site, and it's able to do so because coenzyme A is very similar in structure to acetyl-CoA. If we were to do a Lineweaver work plot analysis where one of acetyl-CoA concentration is plotted as a function of the 1 over initial velocity and analyze this in the presence of increasing amounts of coenzyme A, this generates an intersecting line pattern, which is indicative of a competitive inhibitor. Feedback inhibition is a special type of product inhibition. And so the inhibitor is not a direct product of the enzyme. Instead, the end product is that of a multi-enzyme pathway, where that end product binds and inhibits an enzyme at an early step in the pathway. So it feeds back and inhibits that first enzyme. So let's take, for an example, threonine, which is a substrate for enzyme 1 of a multi-enzyme pathway called threonine dehydratase. That threonine is converted by threonine dehydratase into a product A, and that product A is going to serve as a substrate to another enzyme in the pathway. And so this keeps going until we have a final product, isoleucine. So this is a final product in a multi-enzyme substrate product pathway. If there's too much isoleucine around, it's going to actually come back and inhibit that first enzyme, threonine dehydratase, in this multi-enzyme pathway. So this is a very effective control mechanism. The entire series of reactions shuts down when an excess of final product exists, preventing accumulation of intermediates in the pathway as well as wasted energy. This is a common type of regulation that's seen in metabolic pathways. Allosteric enzymes can be regulated by allosteric effectors. So binding of a small molecule or an allosteric effector causes a change in the enzyme shape and therefore its enzyme activity. Allosteric effectors can be positive effectors, so they're going to act as activators, or negative effectors, so they're going to act as inhibitors. This is not the same as competitive or uncompetitive binding. It's much more complex than that because these enzymes do not fit the Michaelis-Menten model of enzyme behavior. Aspartate transcarbamylase, or ATCase, is an example of an allosteric enzyme. This enzyme catalyzes the condensation of aspartate and carbamyl phosphate to form carbamyl aspartate. This carbamyl aspartate undergoes a series of other enzyme-catalyzed reaction, which then is converted to cytidine triphosphate, or CTP. So carbamyl aspartate is a precursor of CTP, where CTP acts as a negative allosteric effector of ATCase. ATCase is an allosteric enzyme where the rate increases as a function of increasing substrate. This results in an initial velocity curve that is sigmoidal rather than hyperbolic at increasing concentrations of substrate. And so the sigmoidal curve is indicative of cooperative behavior of the enzyme. This is a very distinguishing characteristic of an allosteric enzyme. With allosteric enzyme, there is no Km. This is because the enzyme does not have Michaelis-Menten behavior of enzyme kinetics. Instead, it has a K.5, which is the concentration of substrate at 1/2 Vmax. Aspartate transcarbamylase has a total of 12 subunits. The catalytic subunits consist of 6 different protein subunits that are organized into 2 trimers. So these are blue and purple. There are regulatory subunits, which consist of 6 protein subunits that are organized into 3 dimers. So these are red and yellow. ATP and CTP, which are allosteric effectors, bind to the regulatory subunits and activate or inhibit the enzyme. CTP is a negative allosteric effector, so it's an inhibitor of ATCase. It is one of the end products of the metabolic pathway that ATCase is a part of. So there's actually logic in the way that CTP regulates ATCase. If CTP is abundant, then there's no need to continue to make CTP. The pathway is slowed down by CTP inhibiting ATCase. ATP is a positive allosteric effector, so it's an activator of ATCase. High ATP indicates the cell is metabolically active. Because it's more metabolically active, the cell needs more nucleotides, and so it needs to make more CTP. Let's look at the initial velocity curve of ATCase in the presence of CTP or ATP. In the presence of inhibitor, CTP, the curve is shifted to the right, so higher substrate levels needed to achieve the same rate of the reaction. In the presence of activator, or ATP, the curve is shifted to the left, so less substrate is needed to achieve a higher rate. Enzymes can also be regulated by covalent modification. Covalent modifications are additions of functional groups or other small proteins to an amino acid residue within a protein or enzyme. This is also called post-translational modification because these modifications occur after translation. Many different groups can be added, but the most common are phosphorylation, acetylation, and methylation. Acetylation is the addition of an acetyl group to the amino group of a protein. And so this can occur either at the N-terminus or at a lysine. A great example is lysines on histone proteins can be acetylated. So then an acetyl group can be transferred to the amino group on a side chain of lysines on histones, making acetylated protein. And so this acetylation acts to neutralize the positive charge on lysine. Methylation is the addition of 1 to 3 methyl groups to the amino group of lysine or arginine. This increases the hydrophobicity of the modified lysine or arginine. That is, as we increase the number of methyl groups, this increases the hydrophobicity. And histone proteins contain many lysines and arginines, so they too are also commonly methylated. So I want to take a moment to talk about histones and chromatin. Eukaryotic DNA is wrapped around histones to form chromatin, and the organization of chromatin controls the accessibility of DNA and whether that DNA becomes transcribed or not. And so when chromatin is in sort of this closed, very tight knit conformation, it's actually very repressive to transcription, and so genes are often turned off. Post-translational modification of histones, such as acetylation, can have the effect of opening up the chromatin such that it's more conducive for a gene being expressed. And so when, for example, when histones become acetylated, this causes the chromatin to decondense, which then allows that gene to be in a more open conformation to be able to be turned back on again. Phosphorylation is another type of post-translational modification. Proteins can be phosphorylated on amino acid residues that contain hydroxyl groups, such as serines, threonines, and tyrosines. And ATP is the source of a phosphor group. ATP is reacted with a protein that contains a hydroxyl group, and then this results in a phosphorylated protein and ADP. That presence of a bulky negatively charged group can change the conformation of a protein and its interaction with binding partners. The phosphorylation can have the effect of turning an enzyme on or off, depending on the particular conformation shift that phosphorylation at that specific site causes in the enzyme. The enzymes that catalyzes phosphorylation or dephosphorylation are called kinases and phosphatases. A kinase is an enzyme that catalyzes the transfer of a phosphor group from ATP to some substrate, which is usually a protein. And so when it acts upon a substrate, we say that it phosphorylates it. And a phosphatase is an enzyme that catalyzes the removal of a phosphoryl group from the phosphorylated protein. So when it does so, it dephosphorylates that phosphorylated substrate.